Okay. This is our first exam question. I've picked up three questions that I'm going to go through in today's lesson to summarize what we have looked at, all about mitosis. There was a lot that I couldn't do because we've not had a look at the mitosis practical yet. A lot of mitosis questions have the mitosis practical also involved within the question at some point. But we're going to have a look at these three questions because these three are common as well. So it says animal cells replicate during the cell cycle. Figure 2 shows one animal cell at a stage of the cell cycle. Identify the stage of the cell cycle shown in figure 2. Explain the appearance of the cell shown in figure 2 and do not refer to organelles in your answer. So as you can see, we can see that the cell membrane is dividing and the cytoplasm is also dividing. So we're basically at that stage where we're going to form two identical daughter cells. So that's basically, it's a cell cycle. It doesn't say mitosis, so it doesn't have to be a stage of mitosis. Um, it's a cell cycle. And remember, in the cell cycle, there are three stages. You've got your interphase, you've got your mitosis, and then you've got your cytokinesis. This is going to be your cytokinesis. Cytokinesis, because your cell membrane is dividing and your cytoplasm is also dividing. Okay, we need to explain the appearance of the cell. So as we can see, we can just say that the cytoplasm is dividing. Okay, that's what we can see. We can see that the cytoplasm and the cell membrane are dividing. Okay, so that's why I would say for 2 marks. If I show you the mark scheme to this answer, as you can see, it says cytokinesis. It does allow telophase, so this was a generous mark scheme, so it's allowing you to say telophase. And the reason why is because cytokinesis and telophase are right next to each other. In both versions, you're forming sort of two nuclei. And as you can see two nuclei in the cell as well. That's why it's allowing your telophase as well. It says division of cytoplasm, which we said. You could have said pinching of cell surface membrane, or new cell surface membrane also forming. Chromosomes not visible. As you can see, the chromosomes are not visible. And in telophase and cytokinesis, your chromosomes are going to sort of uncondense and go back to become invisible as well. So if it needs to go through mitosis again, then it can condense again to become visible. Okay, but if you've said cytokinesis and what cytokinesis actually does, you would get your 2 marks. Okay, the next question, it says describe the appearance and behavior of chromosomes during prophase and during anaphase of mitosis. Okay, so remember, we're talking about mitosis this time, and the acronym, we've got your PMAT, you've got your prophase, um, and your anaphase over here. And remember, in prophase, what did I say? The two key parts, your chromosomes condense and become visible. Okay, that's what you need to say, and that's going to be worth 2 marks in an exam. So that's exactly what I'm going to say here. Chromosomes condense and become visible. Okay, so your chromosomes condense and become visible. You could also say that the centrioles release spindle fibers, the spindle fibers attached to the chromosomes. But I'm not going to say that. I'm just because you've said chromosomes condense and become visible. Then you've got your anaphase. Remember, anaphase is the retracting stage. Okay, so in anaphase, you've got your centromere, which is attached to your spindle fiber, and your centrioles have started to retract. So your centromere is going to divide, and your chromatids are going to be pulled towards opposite ends of the cell. Okay, so your um, centromere divides, and your chromatids and each individual chromatid is pulled to opposite ends of the cell. Okay, that's what I would write for a question like this. Um, it seems 4 marks. I've not covered all the lines, but these are the points that they're looking for in an exam. Okay, try being as concise as you can be. So it says chromosomes condense, chromosomes become visible. That's 2 marks there, which we both ticked. You could also say chromosomes attached to spindle fibers, or chromatids attached to spindle fibers, or centromeres attached to spindle fibers, anything like that. But with prophase, these two are always going to be worth 2 marks. Condense and become visible. Those are the two key stages of prophase. In anaphase, the centromeres divide, okay, and the chromatids are pulled towards opposite ends of the cells. That's what happens in anaphase. That's the retracting stage of anaphase. Okay, so that's 4 marks there for a question like that. Okay, and finally, we're going to finish off with a mitotic index question. So over here it says the diagram below shows the student's drawing of one field of view. Name the stage of mitosis shown in cell G. Explain the appearance of this cell. So as you can see in cell G, we can see that the chromosomes are retracting towards opposite ends of the cell. So this has to be anaphase. Okay, so we've got anaphase because the chromatids are retracting towards opposite ends of the cells. Explanation would be just that the chromatids have been pulled towards opposite ends of the cell. Okay, and then it says use the diagram above to calculate the mitotic index for the cells in this field of view. So remember, I said step 1, count the number of cells that you can see. Okay, so we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39. Okay, so we've got 39 cells in our view. Then we need to see how many are in mitosis. Count the number in mitosis. So as you can see, this one is in mitosis, 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 and this one is in mitosis as well. Okay, so we've got 1, 2, 3, 4, 5. 5 are in mitosis. Then we just do the number in mitosis, which is 5, divided by 39. And I'm just going to leave my answer as a decimal. Because another thing, if you wanted your answer as a proportion, then you would multiply by 100. Always safe bet over here. Keep your answer as a decimal. Okay, don't change into a percentage or anything. Just keep it as a decimal. Because some mock schemes I have seen in the past, I'm not sure if this one will say it, but some mock schemes have said reject percentage. Okay, so just keep your answer as a decimal in all cases. Okay, because some will accept percentages, some won't. Just keep your answer as decimal in all cases. So 5 divided by 39 gives you 0 0.128205 and so on. Okay, so I'm just going to round that to 2 decimal places, 0.13. If I show you the answer, anaphase, as we got, chromatids move to opposite poles of the cell, and 0.13, which we got. And this one, it says reject as a percentage. So just keep it as a decimal, don't change it into a percentage, just to be safe for every question. Unless the question says give it as a percentage.